Here we are with chapter number 3, Amino Acids, Peptides, and Proteins. We've got some learning goals for chapter number 3, Amino Acids, Peptides, and Proteins. Well, as you would expect, we want to talk about amino acids. We want to talk about structure and properties of peptides, ionization behavior, and methods to characterize. And these are in sequence as we will cover them, and they're kind of in sequence in terms of importance. And the cool thing about this is once you know the first one, if you know the structure and naming of amino acids, then you're going to be able to build upon that and go on to the second one, properties of peptides and everything like that. And then once you know the peptides, then you can talk about the ionization. It's great. At any rate, we're going to proceed. So proteins are the main agents of biological function. They're going to catalyze reactions. They're going to be involved in transporting substances. They're going to provide structure, which we already kind of talked about that with the cytoskeleton. And they're going to be involved in motion, the motion of, well, entire molecules or entire cells. If you talk about like the flagella for a bacteria or something like that. And it's proteins, actin and myosin, that are really involved in that. Or I'm sorry, actin that's going to be involved in the flagellar protein or flagellar motion and everything. All right, so amino acids are building blocks of proteins. Um, Proteins are linear heteropolymers of alpha amino acids. And that term alpha amino acids, it's kind of funny because in biochemistry, we're always certain to use this term alpha amino acids, but I gotta say, um, let's see, I got my undergraduate degree in 2006, and that was in biochemistry, so maybe I've been a biochemist since then. Um, as a biochemist, I have never talked about a beta amino acid, so it seems a little bit unnecessary. But I think we do that just for like the chemist in the audience who are like, oh well, that's no, the organic chemist, I should say. And I mean no disrespect to any of those people or anything like that. At any rate, here we go. So amino acids have properties that are very well suited to carry out biological functions. They have the capacity to polymerize. They are useful as acids and bases, and they have varied physical properties individually, and they have varied chemical functionality. Okay, so where we're going with this is amino acids are, whenever we use that terminology, and heck, look at this, we were talking about alpha amino acids. We abandoned the discussion of alpha. We went straight into amino acids. I just, yeah, I don't know. Amino acids, which are alpha amino acids, because as we see right here, just a second, my tablet's not plugged in. Now it, no, gotta do the USB dance. Flip the plug. There we go. Now it's plugged in. Okay. We do have an alpha carbon, hence the alpha amino acid. Okay, so whenever we talk about alpha amino acids, really all that we're talking about is this group right here, that R group. That's the only part of an amino acid, almost. That's really important, especially when you talk about protein structure, because you lose a little bit of the importance of the other parts of it. So for our alpha carbon, we have two different groups attached to it. We have an amino group, or this is commonly known as the N, that's supposed to be an N. I'll scribble that out. N termini. And this carboxylic acid is our C termini. Now, one thing that I'd like to draw your attention to is we've got a carboxylic acid that's been deprotonated. We've got an N termini that's protonated. Do you think that that protonation state is going to be important? You betcha it absolutely is. And is this accurate? Would an amino group be protonated when a carboxylic acid is deprotonated? Yes, it would. This is probably drawn at a physiological pH, a pH at 7. The pH was going to be below the pKa of that N-termini and above that carboxylic acid, so the carboxylic acid's deprotonated, the N-termini is protonated. But we move on. Okay, so amino acids have three common function groups attached to them. We already talked about those. The acidic carboxylic acid on the C termini, I'm losing my cursor, there we go, our acidic carboxylic group, or our carboxyl group, our basic amino group, an alpha hydrogen, and then that fourth group, that substituent, that R group, which is going to determine the amino acid that you're talking about. Now, in the case of the amino acid glycine, which is the simplest amino acid, the fourth substituent is also a hydrogen. Okay, so the amino acid glycine looks a little bit like this. C, C, O, O, O. And I'm drawing this out of physiological pH. I'm including my charges. There's the three components that are common to every single amino acid. And then, there we go. There's glycine. Dunzo. All right, we proceed from there. Amino acids are going to be chiral, except for glycine because of the fact that we have two identical groups. This shows us displays of L-alanine and D-alanine. So proteins are only going to include those L amino acids, though. That's something worth noting about them. So the kind of biologically relevant form of an amino acid is the D form of it. Or sorry about that, the L form of that amino acid.